Hello, I'm Kevin, and today I'm going to be talking about the research article, Learning to Match 2D Key Points Across Preoperative MR and Intraoperative Ultrasound. So to give some background, this paper mostly focuses on neurologic imaging. So when they say MR, they're referring to an MRI, which is magnetic resonance imaging. It's done by a MRI machine, which is on the top right picture, and it uses a very strong magnet, and it can take slices of the brain. So this is typically done pre-operative, which means before surgery, and the doctors can use this information to create surgical plans. There's also intraoperative ultrasound, which is real-time imaging during surgery, and this typically uses water-based gels and high-frequency sound waves, and it records, the probe records the sound waves transmission outwards and then receives them, and this is useful for avoiding critical areas in the brain. So the goal and motivation of this paper was to improve post-surgical results and minimize damage, and to see if there are better ways of identifying different locations within the brain, as well as combine these two different methods of images and kind of match them to each other, so you can tell where one area on one image is to the other area on the other image. MR and ultrasound images are 2D, but they create a 3D view when you combine the slices together. So the study aimed to find the common points between MR and ultrasound, and what this means is looking at this MRI point somewhere on the image, where is this point on the ultrasound? So what is the point associated within these slices on the ultrasound? And we covered some of these topics in 568. So we learned about feature mapping, we learned about different methods for analyzing tissue density versus bone and muscle, and we used edge and corner detection methods. We also learned about feature transforms. So we learned about SIFT, which is a scale invariant feature transform, and we'll cover a little bit more of some other feature transforms that they used. So for their methods, they used a multimodal method, which is a combination of tools, in this case MRI and ultrasound. And the reason this is quite challenging is because the images are so different. So they're different in texture, they're different in what kind of information and volume. So as you'll see in a bit, ultrasound images have a sort of wedge shape due to the nature of how the sound waves are emitted and transmitted back. And MRI images have a more standard rectangular shape that can display the brain. So along these images, there's different spatial resolution, and there's a lot of noise associated with the ultrasonic images. And so they need to fix this or map them using a texture invariant feature descriptor, as opposed to something like SIFT, which is only scale invariant. And they use this feature descriptor to look at temporal changes, so changes over time, structural changes, or appearance changes. So the research is they describe a novel texture invariant 2D key point descriptors, and they do matching by synthesis. So they take, I'll go over some of the steps later, but they synthesize ultrasound images from the MRI, and they use it to train a model that can analyze and match key points. And this is done with supervised learning. So it remains texture invariant, and it's robust to noise. And it doesn't require human annotation, so you don't need people pointing at matching points or a large training data set. But it's basically just training a model that can generate output, and it's human interpretable, so someone can look it over and say, yes, these points match, and otherwise, no. So this is one of the figures included in the paper, and basically it just shows the process. So on the left, A is like the ultrasound images, B is how they make the model, and C is the matching points between the MRI image and the ultrasound image. So basically the steps are MRI and ultrasound images, you want to match the 2D points of interest. You generate ultrasound images from the MRI samples. So not real data, but it's synthetic data created from the MRI. Train the model based on the generated images, and then you use that to detect key points, and then you use the model to test the methods and compare to other state-of-the-art feature transform models. So this is something from the paper. It basically explains the mathematical definition of what they're trying to do here. And yeah, we'll see. The keywords here are matching by synthesis strategy, as well as the similarity functions, which is where they try to compare similar points across the images, and they have a very specific way to define which points are similar and which points are not. So for image synthesis, they use a generated network called MHVAE. I'm not sure if there's another way to say that, but basically what that is a multimodal hierarchical variational autoencoder. So this is a thing that's out there already, and they're using it to synthesize ultrasound images from MR samples. And this is one of the equations that's in the paper. I sin US equals G, and G is the generated network, and it takes parameters of theta and gamma. Gamma varies its texture. Theta is basically pre-trained parameters or the weights. So it generates 28 ultrasound images for each MRI with different variations of theta and gamma. So this is an example picture. So the left is the MRI image, and the right are all the synthesized images. And the last one, all the way on the right, is the actual ultrasound image. So you can see here the middle section. There's a lot of different variations. You know, some of them are brighter than others, but the left and right columns are the real data collected from a patient, like a real person. And everything in between is a generated sample from, you know, their synthesized models. So they use training, so they use SuperPoint and dbScan for training. So it's density-based special clustering of applications with noise. So they really wanted to prevent noise affecting the samples. So basically, you detect key points on the MRI sample, and then you iterate over the ultrasound synthesized images and use SuperPoint to detect key points that are within a 5-pixel margin. So if a point keeps appearing on those images, so within 3 images, then you weigh that location as a heavy or more promising candidate location. And then you normalize the patches with grayscale mean and stuff. But basically, you're learning which points keep appearing at the same places. So they use a architecture and loss function. So this is just something that's already out there. Convolutional neural network, CNN. We did that in project 4. Evaluate loss. You choose a lowest loss for backpropagation. They use triplet loss, which is basically an anchor and positive and negative, so matching and non-matching, so stuff like that. These are all like out there, so you can do more research if you want to. Just search up the keyword. So training and optimization. Some of you may see ADAM, adaptive moment estimation, which SGD. So we've seen some of that in TensorFlow and in class, especially the gradient descent. And basically, training and optimization is trained in 2D, pair slices. Models are based on patients in a 1-to-1 ratio. So they basically normalized it. They had 7 cases with MRI and ultrasounds, and they used matching score, precision, and match points as a metric, a result. So these are some of the results they found. Figure 3 shows the matching points. So the points are matched to each other, and the red dots indicate wrong, and the green dots are right. Looking at the table, the comparison with the other state-of-the-art models, the novel 2D key point detector featured in this paper is significantly better in terms of precision and matching score, and those are very important measures, and the only disadvantage is a decrease in the number of matched points. The definition of precision is found on the previous slide. So that's the ratio of ground truth to the number of patched key points. So overall, this is another example of how technology can help improve medical outcomes, and this has shown that we can do better.